we're going to talk about neuron transport. So remember, the electrical signal on the neural lemma only travels in one direction. That's from the cell body toward the axon terminal. But it turns out that things inside the cytoplasm can move any which way. So molecules that are inside the cytoplasm can move back or forth. They can go from the cell body toward the axon terminal. If they move in that direction, the same direction as the electrical signal, we call that an anterograde movement or anterograde transport. If the molecule happens to be moving in the other direction, up toward the cell body, the opposite direction of the electrical signal, then we call that a retrograde transport or retrograde activity. This can happen either fast or slow. Fast axonal transport happens about 400 millimeters per day. We've got microtubules inside of the axon, and there's motor proteins that are powered by ATP, and those motor proteins will go ahead and drag vesicles or molecules up and down the axon. Slow axonal transport is really, really slow. It's about 0.1 to almost 3 millimeters per day, and this is from the flow of axoplasm. Now, when we look at neurons, we've got several different kinds of neurons. Multipolar neurons are the most common type, and these are the ones that you're probably comfortable thinking of as neurons. We've got a lot of dendrites. They have one nice, big, long axon. But we do also have some other types of neurons. There's bipolar neurons. They have one dendrite and one axon. And we really don't have lots of these. They're pretty limited. We have them in the retinas of the eye, mostly. And then we have unipolar, or what are known as sometimes as pseudo-unipolar neurons, where one process extends from the cell body, and then it splits into two. The peripheral process functions as a dendrite, and then the central process leads toward synaptic knobs in the central nervous system. Some neurons are anaxonic. They have dendrites, but no axons. Here's an example of a bipolar cell. Remember, these are very limited in the body, found mostly in the retina of the eye. A pseudo-unipolar is in the middle there, and then our multipolar cell. Now, we tend to classify neurons functionally according to the direction that they're sending their action potentials, that electrical signal. Sensory neurons, also known as afferent, are going toward the central nervous system. Motor neurons are efferent. They're all heading away from the central nervous system. And all of these are going to be multipolar. And then we have a lot of neurons that are neither motor nor sensory. These are called interneurons or associative neurons. These are going to receive, process, and integrate information from lots of other neurons. So these communicate between the sensory functions and our motor functions. These are all located within the central nervous system, and they're about 99% of our neurons. They're generally multipolar. So here we have a functional classification of neurons, where our sensory input is coming into the central nervous system. And with sensory neurons, most of the somatic ones are going to go through what we call the dorsal root ganglia. So the ganglia is the collection of cell bodies outside of the central nervous system. These tend to be more of our pseudo-unipolar cells. We have our cell body right here in the ganglia, and the dendrite is off to the left, and then the central axon part is to the right. Then we've got a purple interneuron that's hooking together the signal between our sensory neuron and our motor neuron, and then the motor neuron is a multipolar cell where the long axon is going out to the muscle to form a neuromuscular junction. A synapse is a place where a neuron connects to something else. It could be another neuron, it could be a muscle cell of any type, or it could be a gland. Chemical synapses are much more common than electrical synapses. The electrical synapses we do have are where the presynaptic and postsynaptic neurons are tightly bound together with gap junctions. These are very fast, and there is no synaptic delay in passing that electrical signal. Our chemical synapses have presynaptic neurons, where the axon is going to terminate, then the synapse, and then our postsynaptic neuron that's going to receive the signal from the presynaptic neuron, usually with one of its dendrites. The gap between the presynaptic neuron and the postsynaptic neuron is called the synapse or the synaptic cleft. There's a little bit of gel inside between the two neurons, and as we release a chemical from our presynaptic neuron, that chemical is a neurotransmitter. It's going to diffuse across the synapse, bind to receptors on that postsynaptic neuron. That's going to initiate some kind of postsynaptic potential, what we call a graded potential. And this is very close to the same thing that we had on the neuromuscular junction, but it's a little bit different. It's a little more complicated. And it does take time for all of these things to happen, and we call this a synaptic delay. Now moving on to glial cells, these are what I like to call the roadies of the neuronal tissue. Glial cells are non-excitable. They are nervous tissue, but they're not neurons. These are all the helper cells. They support the neurons in their rockstar lifestyle. They're much smaller than neurons, but they far outnumber the neurons, and they're about half of the volume of the entire nervous system. They are capable of mitosis. They protect and nourish the neurons. They provide physical scaffolding, chemical protection, and they're critical for normal function at neuronal synapses. Some of the types of glial cells we have are astrocytes. These have little processes that end in what we call perivascular feet. Peri is around, vascular is the blood vessels, and so the perivascular feet encompass part of a blood vessel. These are the most abundant glial cells in the central nervous system, and they help form what we call the blood-brain barrier, which is a chemical barrier that limits what kind of molecules can get into the brain. Astrocytes also form structural support for nearby neurons, assist in neuronal development, and they will occupy and fill up the space of dying neurons. Ependymal cells are found also inside of the central nervous system. These line the cavities in the brain and the spinal cord. They're part of what we call the choroid plexus, which produces cerebral spinal fluid. Microglial cells are a type of small white blood cell that wanders inside of the central nervous system, and these are phagocytic, and they're going to help clean up any debris or any infectious material that should get into the central nervous system. And then our oligodendrocytes are large cells with a few slender extensions. Oligo is an interesting Latin root that means basically 3 to 10-ish. So they're going to send out a small number of these extensions, which then are going to partially wrap around portions of axons of neurons and form the myelin sheath inside of the central nervous system. So here we have our neuron, our big soma or cell body, and the axon going down from there. And then here's some oligodendrocytes that are sending out their little processes to help wrap up and myelinate the axon of the neuron. Over here is an astrocyte, and it's sending out these little perivascular feet that are helping to encompass a blood vessel. We've got a little capillary over here. Any molecule that's inside of the capillary, before it can mess with the neurons, will have to get through the cells that make up the capillary and the cell of the astrocyte. Remember, all of your cells, including the astrocytes and the endothelial cells of the capillary, are made out of membranes, and all those membranes are semi-permeable, right? They restrict what goes in and what goes out. This is why if you take aspirin, it doesn't tend to mess with your mind, but if you drink alcohol, it does. That's because the alcohol molecule can fairly easily diffuse through the endothelial cells and through the perivascular feet of the astrocyte and directly affect the neuron, whereas the aspirin molecule cannot pass through. In the peripheral nervous system, we also have glial cells. These are satellite cells and Schwann cells. The satellite cells will help protect the cell bodies in the ganglia and out in the peripheral nervous system, and then the Schwann cells are going to do the myelination of axons that we find in the peripheral nervous system. So here's our peripheral nervous system. We've got our dorsal root ganglia and some satellite cells surrounding the cell body of that pseudounipolar neuron. And then down at the bottom, we have our Schwann cell, and that's just one of them, partially wrapping up the axon, kind of like a pancake around a sausage. Let's take a few minutes and look at a clinical aspect of the central nervous system. Neoplasms, or tumors of the central nervous system, come from unregulated cell growth. Now, this doesn't really come from the neurons. Remember, the neurons don't do cell growth. They don't do mitosis. So if anything does happen, it's going to have to come from the glial cells. So all of these tumors are going to come from tissues with the capacity to undergo mitosis, mostly glial cells, or occasionally from the meninges. A glioma would be a tumor of a glial cell. They can be benign, they can be malignant, and the malignant ones can metastasize or spread.